Earth's coral reefs are not just visually stunning ecosystems. They are indispensable pillars supporting various aspects of human life. Underwater cities sustain the global marine food web and protect coastal communities from severe floods and hurricanes. Economically, coral reefs generate billions of dollars through their protective role, support of fisheries, and attraction of tourists. However, escalating threats posed by climate change are driving worldwide declines in coral reef health. Rising CO2, hypoxia, solar radiation, nutrient availability, and rising temperatures alter the delicate balance within these ecosystems. In the past 50 years, studies suggest that we have lost half of the world's coral reefs. Recognizing the urgency of this situation, recent research has turned towards synthetic biology applications, such as assisted evolution, as a promising solution to restore reefs to their former productivity. Endolithic microalgae and fungi residing within coral skeletons play pivotal roles in capturing light for photosynthesis and providing nutrients to corals as food with carbon sugars. Other fungi may act as guardians by protecting from UV radiation and have also been found to exhibit beneficial antimicrobial properties. Heterotrophic bacteria work as the metabolic engineers of the reef by cycling essential nutrients like sulfur, carbon, and nitrogen, while also safeguarding against certain pathogenic invaders. To control these bacterial populations, viruses are essential for facilitating the transfer of genes and acting as natural regulators of this community. Archaea, particularly ammonia-oxidizing archaea, and their involvement in the process of nitrification, which converts ammonia to nitrite and then to nitrate, is crucial to control the amount of ammonia released through the excretion of waste products by fish and other marine organisms in these productive reefs. The reintroduction of nitrate, which is an essential nutrient for algae growth, can then be accessed by the coral's main primary energy producer, the dinoflagellate Symbiodinium. Stress events can lead to shifts in the microbiome that alter the balance of these ecosystems. Elevated temperatures and rising CO2 may induce increased microbial activity in corals and stimulate nitrogen fixation activity. With excess fixed nitrogen, the endosymbiotic Symbiodinium experiences rapid population growth. This shifts nitrogen limitation to phosphate starvation, and with this rising population size leads to coral bleaching as corals expel their algal partners, and which can lead to widespread disease and death. Despite the challenges in unraveling the complexities of these different players, recent scientific advances in next-generation sequencing technology, such as Metabolomics, have served as the basis of manipulating microbial symbionts as strategies for restoring reefs. Multi-omic techniques of healthy and bleached corals allow for insight into the microbiome metabolic pathways that inform predictions of optimal microbiomes. These microbiomes can then be used as entire probiotics to administer to corals with the aim to enhance certain traits. The second strategy is to create synthetic microbiomes from experimental evolution to manufacture increased stress within symbiont strains to decrease coral bleaching. Through controlled laboratory experiments, isolates can be exposed to long-term stimulated environmental stressors to evolve favorable traits to enhance new hybrid tolerance to stress. Things such as altered gene expression or production of protective compounds of interest that may protect from things like reactive oxygen species. This can be conducted and then reintroduced and tested in the coral host to create more thermally tolerant offspring at various stages of the coral life cycle. Through culturing of certain bacterial symbionts, genome and phenotypic data from cultures can inform genetic engineering efforts. Genetically modified bacteria can then be reintroduced into corals in certain stages of life. Recent advancements in CRISPR-Cas9 genome editing has allowed for the insertions of certain genes and pathways that may confer enhanced thermal tolerance to the host. Strategies include engineering thermotolerance and manipulating metabolic pathways to maintain symbiotic dynamics. While synthetic biology holds promises for coral reef restoration, it presents ethical and regulatory challenges. Careful assessment and containment are essential to minimize ecological risks that could be associated with the deployment of certain engineered microbes. Rigorous testing that includes small-scale experimental systems that are then scaled up to mesocosms which mimic field conditions, are necessary. Growth characteristics, effectiveness, competitiveness, trade-offs, and repeatability for mass application all need to be considered before field application. Although potentially efficient, microbial manipulation is extremely specific, and specific studies and strategies must be applied for each site. The preservation and restoration of coral reefs is paramount for the health of our oceans, all of their organisms, and the well-being of coastal communities worldwide. Through innovative approaches and the help of the invisible microbial superheroes within this complex system, we can find solutions to protect and restore coral reefs for generations to come. By safeguarding these vital ecosystems, we ensure the sustainability of marine food webs and biogeochemical cycles of C, N, S, and P, all essential for human life and ecosystem resilience worldwide.